Sipke Hiemstra, Director for the Center of Genetic Resources in the Netherlands, is going to discuss ethical questions related to cryopreservation strategies for animal genetic resources. Sipke.
Yes, thank you, Harvey. So you all received the questionnaire already. I will give some further background to this questionnaire and the ethical questions that we want to reply to on gene banks and the choice of breeds. So what is the background of these ethical questions? We had as an IMAGE project, we had an ethical review committee looking at the project, and they have asked us as a consortium for a report on the ethical consideration of stakeholders for the choice of breeds to be cryoconserved. So that's an ethical question regarding the ethical review committee. And in addition to that, we work on innovative cryopreservation and reproductive technologies. And you can imagine that there are some ethical questions related to those new technologies. So we have a series of meetings, including this one, where we discuss and consult a diversity of stakeholders on these type of ethical questions. And I will give some further background to the questions. You have seen these pictures in the documents of the FAO for this meeting. Michel also said a large proportion of the breeds globally is at risk. So that's the starting point for our work to conserve. So back to the question, the choice of breeds. The ethical questions behind that. To our opinion, it refers to what kind of objectives a gene bank has. That could matter. What kind of criteria are we using for choosing breeds or for choosing individual animals? You could consider it as an ethical question. Also the question of who shall prioritize or who shall decide on prioritization, on decision made by gene banks. The question who pays is relevant there. And also if you compare in situ and ex situ approaches, there may be a balance or a conflict or interaction between ex situ and in situ support, what you could consider as an ethical issue. Back to the survey of IMAGE on the gene bank objectives. So most of the current gene banks have two major objectives. First of all, to support the in situ conservation of local and native breeds. And secondly, to have a long-term perspective as an insurance policy. So those are the two main objectives mentioned by gene banks. But there are some other objectives also, as you can see on this slide. Then the question of criteria for choosing breeds and individual samples to be cryopreserved. So what are the criteria? You could look at the degree of endangerment of a breed. You could look at different values, economic value, cultural value, environmental values of breed as a criteria to choose breeds. We could look at genetic distinctiveness of a breed. If a breed has more or less within breed diversity, that could be a criteria for choosing or not choosing a breed. A breed may have, or an animal may have special characteristics. Or, that's a more general question, should we cryopreserve all breeds? Because all breeds have an intrinsic value maybe to be cryoconserved. All these type of issues are also in the questionnaire, as you may have seen. Then stakeholder involvement. On the right-hand side is an unreadable slide, so don't look at that. I just want to emphasize that the current gene banks are quite different in terms of organization and the type of stakeholders involved. Usually the government is involved in some way, breeding associations are involved, breeding industry, the gene bank manager has an important role to play, researchers, and in many cases there is a multi-actor board that actually decides about the strategy and about the choices to be made. On the right-hand side you see a slide from the survey of IMAGE, and it shows a lot of diversity in terms of how gene banks are organized and who decides and who pays for the activities. The balance between in situ and ex situ conservation, so we all know, we say that all the time, that they are complementary to each other. Both strategies have pros and cons, but there are differences in terms of responsibilities for the different strategies, and also differences in terms of who's paying for what. Also, the use of subsidies can be used for in situ conservation support, but also for the ex situ conservation for gene banks. And I think for governments it's an important question how to balance the financial support towards the two complementary strategies, ex situ or in situ. Then the second component of our ethical questions is about innovations in cryobiology and reproductive technologies. Well, this is not my area of expertise, but I try to summarize what it is about. If you look at the survey again of IMAGE, then we see that 97% of the germplasm collections is semen. So only 3% is other type of material. Embryos, oocytes, whatever kind of material. I come back to that. Why is that? Because it's technically possible for all species to cryopreserve semen. So, and it is not invasive, or at least less invasive than many other genetic material that can be cryopreserved. At the same time, we know that there's still a need to improve. Michel also explained that, to improve cryopreservation protocols also for semen. But the question is, semen is only the male side. So the question is, are there alternatives or new approaches that we should investigate further or implement further? So there's research in IMAGE going on some new ideas, new technologies, also to conserve the female side mainly, because with semen we have the male side, but we want to also to conserve the female side. And another argument is to reduce the cost of cryoconservation and to make future use more easily. So what are the options? We can look at embryos, oocytes, gonadic tissue, ovarian tissue, primordial germ cells, somatic cells. Those are the additional options besides semen. Well, embryos, it's possible for many mammals, but not for birds. Cryopreservation of oocytes, also again only for mammals, in particular for humans, it's a realistic strategy at the moment, but not for birds. Gonadic ovarian tissue, it's an interesting approach for birds, but also for other species. Primordial germ cells, also an interesting strategy to conserve the female side or to conserve birds in general, but also for other species this is an interesting, new, innovative technique. And then we have somatic cells. You can store skin cells, somatic cells of any species, but then you should realize that future use is related to cloning or reprogramming methods of cells. So I will highlight the two main components of IMAGE. So it's about gonad transfer. So if there's no other method available, then gonad transfer could be an option for birds in particular. And you could cryopreserve already material from very young animals, or you can cryopreserve even material from dead animals. And there are different options. I will not go into the detail what kind of cells, what kind of material you could cryopreserve. This is a slide of Elisabeth Blaise-Bois from INRA. She is the leader of this work package in IMAGE on reproductive and cryopreservation technologies. I'm not an expert, but you can, if you look quickly at this slide, you see that you can isolate tissue, you can cryopreserve gonad tissue, and then this could be followed by grafting, transplantation of this material in another animal. And in this way you can conserve and use later the material of the original animal. Well, looking at the ethics and welfare issues, what is the, besides the very nice, interesting future ideas, surgery is needed for this type of approach. There's a risk of infections, and you need very specific skills to do this. It's not like semen. that you can collect semen easily and freeze it. You need very specific skills here. Another issue here is that you need to sterilize the recipient animal. And there's a problem of immune compatibility between the donor and the recipient animals with this type of technique. And you need immunosuppressors. So quite a list of issues. You have to realize that this is associated with cryopreservation of gonad tissue. But technically it becomes possible. It is possible. Some results have been shown already. So the question is, do we want this as an alternative approach for, in particular, birds? Then we have primordial germ cells technology. That is, the primordial germ cells are the precursor cells of germplasm, female side or male side. And you can isolate those very early precursor cells and cryopreserve them, and then introduce them again in another animal. And this animal will produce genetically the germplasm of the original animal you isolated the material from. So it's an alternative also for semen and embryos, and it's very quick. You can, in theory, you can have only one generation to be able to recover a full donor animal genome. But at the moment, results are still very variable. And from the ethical side, you need sterilization of the host animals, or you have to create a transgenic, sterile host animal using genome editing. It is invasive, so that raises some welfare issues. And at the moment, it's still expensive, but we may expect some rapid developments also in this area. So you all have filled the questionnaire maybe already before the meeting. You can do it again now if you want. So we have prepared a list of questions, a very simple list of questions, to get an idea of your of stakeholders' views on the different questions I raised here in this presentation. The questionnaire will stay anonymous. You don't have to put your name. Sorry? You should not put your name. So this is the end of my presentation, and I hope it's a good start for further discussion now.
And thank you both for those great overviews. Anyway, we'll open it up for questions. Does anyone want to kick this off, please?
Yeah, thank you, Professor. And thank you, the presenters, for the insights into the gene banking. My name is Charles Lagu, the National Coordinator for Republic of Uganda. And I'm here with my minister in charge of animal industry. Under the African Union Bureau, Uganda was selected to host the regional gene bank. So we are just in the early stages to start the regional gene bank, mainly to take care of the indigenous breeds in that region. About 12 countries of Eastern Uganda, or Eastern Africa, and IGAD countries. Now, my issue is how can we cooperate in these areas now that you have made some effort in this, so that we get synergy, we get cooperation, to make sure that the regional gene bank in that part of the region is kickstart, and it is in line with the acceptable standards. That is my question. Thank you very much.
Thank you very much, Charles.
Thank you very much for this question. And indeed, I think it is a very important initiative in Africa to have these regional gene banks. So the first answer would be that in our Work Package 7, we are going to organize a training session and meetings open to non-partners. And so I think it would be important that we get your address so that we can invite you when we set up such meetings and seminars. Of course, all the new methods or tools will be publicly available. Well, when it is finished, it is published. So when you attend a seminar, you can get the information a bit ahead of time, but sometimes it is not completely validated, so it's better to have the thing published. And I think because FAO is partner of IMAGE, it's very important for us to use the FAO framework to get in touch with many countries. That's why we have this side even here. But for instance, typically the survey ongoing for quality management is a way to understand the needs or the real the major blocking points. Or I discussed well, 2 years, maybe 1 year and a half ago, we received in my laboratory the colleague who is in charge of the Western Regional Gene Bank in Africa and Burkina Faso. And so he visited all our different structures, and we worked with him on the different steps from the sampling to the storage to the distribution. And obviously the animal health issue was predominating in the discussion we had with him. But all this question of a unique identifier of an animal, also sometimes it seems quite technical or material, but it can be limiting. So all this checklist we can share with you.
Much to add. I think in general there's a lot to gain still by exchanging protocols, exchanging knowledge, exchanging the way we work between countries and also between regions. So I would say we should continue to do things like this and set up collaboration.
Several of us were involved a while ago with a workshop on gene banking, animal genetic resources. We might ask whether this is a good time to do something similar. Jamali had a question.
Jamali from Tunisia. I would like to thank the two speakers. It seems to me that image is a misleading word, because you are managing genetic material, but you are not managing the animal genetic resources. You are not managing through the main actor, who is the breeder or the livestock owner, where you can have records on your animals and you can manage if they are well selected, if they are well fed, if the reproduction is right. So this is the management, I see it. But in here, you are out of the picture of the man. You are just managing biological material. And for this case, I am a little bit puzzled with image as a word. Thank you.
Okay, thank you very much.
First of all, we should not be prisoner of a word. You know that when you prepare such an EU project, you always have the hunting for an acronym. So we need to think of a short word that you can remember. So at least you remember it, even if you disagree with it, you remember it. So, and it can be, you don't need to translate it, image or image. And what is innovative is that we want to use the gene banks better for the entire population. So that's why the aim of the Work Package 1, the one that is called involving stakeholders, that's typically what you say. That's the place where we need to work with the users, the breeders and the farmers, but then it is very difficult to organize the representation of these people. Because since we have several species, it was also one choice. We could say we take maybe only one species, and then we better involve the people in charge of the breeding program on the field. But we thought that gene banks are always multi-species, and so we needed to keep this dimension because it is also enriching, because some solutions can be found in some species, and well, all the colleagues always find it is interesting to compare situations between species. But really the issue is to involve the stakeholders, including those who are in charge of the management on the field. But the main point, because this was the topic of the call, was how to manage gene banks for a better use of genetic resources. So that's why we took this approach. But I agree, it is not the single approach that can be taken.
I might just add, if I could, if we go back to one of Michelle's early graphs that she showed, the Venn diagram, this overlay or the intersection between in situ, ex situ, I think is an important point in trying to advance that interaction. Sam?
Thanks, Harvey. I'm Samuel from Brazil. I have two questions, if I may. The first one, congrats for both talks. The first one, I would like to know a little bit more, what's the role of South America countries in the IMAGE project? Are you more exchanging information with them, or more best practice, or more supporting the growing and the training of their gene banks? And the second question is, how is the improvement of your honeybees and aquaculture collections? Are moving fast, or still technical problems? Thanks.
The countries from South America, so they are involved in the training work package, and but they also attend our main meetings in Europe, and it depended on the stage of maturation. But for instance, for Colombia, for Costa Rica, it was important to improve the genotyping data on their. They have both an in situ conservation flock, which is an original point, and frozen stock, but they have this. They have the on-field population, this in situ conservation flock of 4 population, and the gene bank. And so we will fund in this project additional genotyping to guide the sampling from their conservation flocks to the gene bank. So they are really the laboratory work supported by the project. It is not only traveling. And for Argentina, it was not their request. They did not put that in the first, so that's why it is not included for them. But if we look also at Morocco, Morocco had already participated in a European project with a lot of genome sequences on sheep, and so we are going to include their data with new data produced on Europe to make this geographical gradient analysis of sheep diversity from Morocco to Northern Europe. To have a better comparison. And so the second question was already the honeybees. So honeybees, it was a discussion at the preparation of the project. Several of my colleagues, at least in France, we were in favor because we have a small image, I would say, called CRB project in France, where we are working on honeybees, biotechnology of conservation, and typically to try to conserve the embryo of honeybee, and not only the semen. But adding the species did not make consensus during the preparation, and also if we have more species, we do less on each. So finally it was decided not to keep honeybees because maybe only 2 partners out of 20 were really motivated to work on them, so there was not a big added value from the consortium. And fishes, it was very simple answer that there is an infrastructure project called Aqua Excel in Europe, which is networking all the experimental facilities for fish populations. And in this project, they are also working on conservation of cells for this line. So it's not exactly the same, but we said we should have collaboration with them and not be redundant. So, but you're right that there are also technological developments for conservation of fish cells and fish semen ongoing in Europe. Yes.
Okay, next question.
Thank you very much for the organizers of this very important event. I'm representing Sri Lanka here, based in the Embassy of Sri Lanka. I have two the questions for clarification, mainly for the coordinator of image. And I understand that, you have around 13 partner countries. So most of them are from the European area, and then 4 other non-European countries. So my first questions for clarification goes really now how the other countries, maybe the countries like actually my country, how can be a partner country with the image. I wonder, my knowledge is very poor, whether you have any ongoing programs with the countries like Sri Lanka and neighboring countries in South Asia in terms of gene bank. So basically I would like to know that how and what would be the procedure like. Is there a particular procedure to be partner with image? The other questions really, I understand that you are trying to make the awareness, right, about these innovations and the way of gene bank. So apart from that, I want to know really whether you are managing, you are keeping up also, you are how can I say, maintaining the gene banks as well in terms of the genetic resources, other than you are helping with the innovation, whether you are going to maintain the, how can I say, the properties of genetic resource. I hope it is clear really. Thank you. Yeah.
Thank you.
Strategic questions. And so regarding partnerships, so there is a mechanic in EU project. Once it has started with a list of partners, it's very rare to include new partner. It is not impossible, but it is a lot of bureaucracy and negotiation to modify the grant agreement. So I think the answer is rather to invite other countries to our seminars or training session. And as I said for Uganda, that's why we have FAO on board, so that they can help us to have the contacts. Otherwise, we would not know everyone. And I don't know whether there are similar programs or research agencies in Southeast Asia, but one idea also would be to promote similar projects in other regions. If we can show it is useful and it is working, then it can be done in other contexts. And for the awareness, for many of the partners of so we are 13 European countries, 4 non-European countries, so 17 countries for 28 laboratory partners. And several of the partners are themselves managing a gene bank. So they are doing research, but they are also involved into the operation of their national gene bank. So we are connected with the real world. But the aim of the project is not to support their daily operations. It's to propose new ways or new tools to facilitate operations, but where the project is not supporting the current operations of the gene bank. But we benefit from the experiment of the people managing gene bank, because we have partners from Netherlands, France, or Austria, and also Italy, who are involved in their national gene bank.
Carl from Canada. I just want to say first, a great presentation. I really enjoy it, but I have ethical questions for you. How, in your Venn diagram where you show the producers and the ex situ, in situ conservation, how do you deal when you have ethical issue faced with a breed where the standard traits have not been defined very well, and how you can use reproductive technologies to help them to manage their resource? When you have a fight inside the association to say this is the true purebred versus the other one. The other question related similar to that, how do you deal with culture when reproductive technologies that you can use to manage resources are not welcome for some community?
Thank you.
But that's exactly the question. In the project, we are not funding collection enrichment. We are funding the development of tools or new data. But the Commission insisted about this ethical consideration, which they considered it is in our field, although we are not giving advice to the colleagues on that in the project. So, but ethics is more questions than answers, as you may know. So I agree that, well, what we are working on is not to define the perfect animal that has to be conserved because it is the perfect one. This can be a question to discuss. It was not one of the options of the motivation to store ideal animal. We did not. So you can add other motivations for storage, but the main concern within the project is that we are supporting or funding the development of tricky technologies, and we need to know ahead of time if these technologies will never be accepted. We need to put the money on something else. Otherwise, we fund a fancy technique, we have a fancy paper, but it is not used. So, and the colleagues are not always so ready to accept that. The colleagues working on gonad grafting in Hungary, they are fascinated by their biological model. They are very happy to do surgery, to have success. And when we tell them at our annual meeting in Germany last year, we had one of our members of advisory board said, but is it ethical to do surgery just for conservation of genetic diversity? And they were surprised by the question because for them it was not a question, but for others it is a question. So that's why the Commission has been challenging us as the whole to make a report of the opinions. So we will not make a conclusion, this is what we should do. But the Commission wanted us to collect different opinions and to make a report explaining the different positions. And myself, I am concerned about the use of the money of the project. If we use it for a technique, that would never be used. So, but that's why we are in the middle of the project, so it's not...
In particular, when you look globally, it's of course very much culturally dependent what the answers will be, also within Europe, but if you go globally, it's even more diverse. So there's not one answer to the question, but we have to make visible what is the diversity of answers you get on these type of questions. So I could give the question back to you. What is your opinion? So you raised the question, but...
No, that's totally, I raised the question because even myself, I do face this situation sometimes, and this is a debate that I start with the associations, which I launch to them this question. Yes, okay, but are you ready to lose the breed if we don't have technologies and etc.? And we try to work together to define this. However, it's not an easy question, especially when you face with an issue where you have a person that do not believe in reproductive technologies. Even you have probably a lot of my colleagues remarked that the reproductive technologies research funding is not a sexy thing right now to request funding on this. It's rare, it's hard to get funded to do those ones. So it's become more a larger debate at this moment.
Thank you, Carl. Maybe go ahead.
One point about this, because this was discussed at the preparation of the project. Some partners said, but why do we give so much money to the work package on reproductive biotechnologies? And my answer was that we encourage many governments to fund just semen, even frozen semen. And what is the success rate we can promise for a range of species? For cattle, there is a lot of data. But for other species, so for chicken, it is entirely variable. For sheep, at least with the current technology, you need some surgery, or it is a not so routine technique. So that's why we need to also improve the techniques if we want the gene banks not to be sometimes what I call a cemeteries of cells. Because if we are happy, we have ticked the box of the FAO survey, I have many breeds in my gene bank, and if they never come out of the gene bank, what is really the usefulness for the strategy of your country? That's really my motivation for this project, to raise these issues, because a gene bank has to be dynamic. And if it has to go out, we need to get good techniques to recover some material from the gene bank.
Maybe just a follow-up question. You mentioned a lot of this was being driven by policymakers kind of asking you the question. When you go to your stakeholders, livestock producers throughout Europe, and you ask a similar set of questions, particularly like, okay, do you get the question, why isn't my breed in your gene bank? What are some of their responses to these types of ethical questions?
We are going to organize a specific meeting for this in October with the stakeholder of the French Gene Bank, because if we just put on the screen, we will have a discussion on ethical issues. Maybe they do not come first of it. They say, oh, but is it useful to me? I have something else to do. So we have to organize a stakeholder forum there where we explain the new results we have, new methods, and then we include the ethical session, because it is not yet in the everyday life to have an ethical debate. But I anticipate very different answers, because I take one example in France of a breed which was almost disappeared. There were 2 animals remaining. So I am a geneticist. When you have 2 animals remaining, for me it is lost. But the breed association didn't want to admit the breed was lost. So they were happy to find a colleague making a clone, to have at least a new animal, a 3rd or a 4th animal of that breed. And maybe if I had interviewed this person 5 years ago, what about cloning? I say, oh, no, it's very bad. But at the point that suddenly realized that the breed was going to disappear, then they asked for it. And so we said to our colleagues in reproductive biotechnology, but you are just postponing the disappearance maybe by 5 years because 1 animal more. But he was so happy to show that the techniques could be useful. The farmer was happy. And so we have a frozen semen from a cloned bull in our French National Gene Bank. Nobody is asking to use it at present. So you can have this sort of, that's what is typical for survey in the public. There is an expression I like very much in social science. You have the declared preference and the revealed preference. So the declared preference is what you will answer to a survey. And the revealed preference is what you are doing, and that can be observed on objective data. And sometimes it differs, and sometimes it doesn't differ. So when it differs, then we have an issue, because we get answers to surveys that may not correspond to the real behavior on the field.
Thank you. I'm Nina from Norway. I want to follow up your talk about the questions and reality, because I find that especially question number 3 and 4 in your survey very difficult to answer, because they are not precise. And I guess you interpret these questions different to me. So then, of course, it will not correspond with reality. So it's not only that I would choose different when I read the question and then when I maybe in a real situation, but it's also that we interpret the question differently. So to me, I've been working even more with the questions in the questionnaire, because some of them are, I feel, kind of impossible to answer because they are not accurate. So that's also a challenge with surveys.
The survey, there is a free text area. So if you think that you want to clarify one point or we missed one issue, that's why we added this free text.
Thank you. Firstly, I'd like to congratulate you for the work that you have done and for the presentation. I just want to ask a question. Since coming from the Oceania region, it's very different from this part of the world. Bridges that are supporting the existence that are culturally important to the livelihood of people within the region and also maybe in some other parts of the world as well. Is there any study or baseline that provide the platform that will direct some of these conservation practices or conservation technologies that we are using to ensure that bridges that are important for the existence of human race in that particular region are also taken into account when we in this type of breeding programs that like you mentioned in your presentation. Thank you.
Of course, I do not know well the background in your country and how breeding or farming is organized and what can be the added value of a gene bank, because very often starting with a good management of in situ populations or live animals is the first important step, because we cannot set up a gene bank if we do not have a well-described situation of the livestock. Or you can, the other option would be to sample randomly, but then if we do not know what you have in the gene bank, who is going to use it? That's always the main question, that if we don't get enough information, if you take samples in emergency and you do not collect information, well, the motivation to go back and use it will be low, and so maybe it's not useful to proceed that way. But I think there are already guidelines from FAO, so we have to take that into account when updating the guidelines. That is one of our deliverables at the end of the project.
Okay, some other questions. Charles has another follow-up question.
Thank you very much for the opportunity. I would like to request the presenters to consider the policy aspect of the gene bank, ethical and technical wise, because particularly for the in situ conservation and hinging on indigenous breeds, they are dangerously threatened. Why? Because the strive is to go for more productive animals, and therefore threatening the indigenous animals with the specific desired characteristics. Either they are resistant to disease, either they have a unique trait, or they have a cultural value, and so forth. So for those areas, at farmer level, public institution level, they are dangerously threatened, because the desire will be to go those with high productivity. So we need a balance, because we need both the in situ and ex situ. So where do we strive this in our member countries? So I thought that is an important issue, so that from policy perspective, from advocacy perspective, the two coexist. Yes, we need the ex situ, we need the in situ, but we have this threat, other than the animals, but also the land is threatened. So I thought that is an issue which we needed to, it should be a point of concern Thank you very much.
Comment, and I think there was one, I don't know whether it's policy or ethical issue, more than 10 years ago when there were these large-scale programs of storing a piece of air in liquid nitrogen and telling to the government, oh, you're safe, you have saved the diversity of your country because you have all these skin samples into liquid nitrogen, and there was a risk to stop supporting the farmer program because of this reason, which is not a good reason, because it's not because you have cells into liquid nitrogen that you should not care of the proper management of the populations in the field. So that's one message, or we can illustrate it.
Yes, I also fully agree. I think in general, national policies tend to favor mainstream activities. I have some examples in our country. So you need specific policies or derogations or specific measures to also look at the in situ conservation of less mainstream activities. That's my opinion.
Thank you. Maybe let me just, in the few remaining minutes, among our gene bankers in the audience, why Sipke had a list of 4 or 5 items there that were kind of this, fell into this ethical issues, who chooses, who pays, protocol use. Just kind of by a show of hands, why are you all experiencing similar kinds of questions? Come on, raise your hand. You know, don't nod. Carl? Okay. Uganda has some gene banking as well, yeah? So you're seeing these kind of questions. Okay, very good. Korea, South Korea as well. Yeah.
Okay, thank you very much. In Korea, we have separate 2 sectors. One is a private sector. It's mainly widely used in corporation organizations. They use in gene bank. Then the other one is the government sector. It's a long-term conservation purpose. Okay, we have 2 different systems. Thank you.
Thank you very much. We're at 2:30, which is our scheduled time to conclude. First, I want to thank Michelle and Sipke and Tessa, who organized all this, for bringing us together so we could have this opportunity to hear what's going on in IMAGE. And we appreciate your participation, your comments today, greatly appreciated. So thank you very much.